what I wanted to talk to you all about today were some Huntington's disease research and clinical advances. We just came from a Huntington's disease sort of clinical research update up in Nashville. There's some really exciting news coming out, and I think some of you have actually heard some of the exciting news from some of the research trials that came up, particularly the AMT 130, and I'll discuss some of those things a little bit later today. But I also wanted to give you a little bit more of a flavor for Huntington's disease and some of the other advances that have gone into it. And hopefully this will bring up some other questions that you might have, but also provide you some new insights in the disease. It's really an exciting time. A lot's happened since the discovery of this Huntington's disease and the discovery of the gene. So I'll go through that. I probably don't need to go over this for those of you who have Huntington's, but I'll just talk a little bit about what Huntington's is. And so this is a progressive degenerative neurological disease that's characterized by sort of this triad of symptoms. So the triad of symptoms that include abnormal involuntary movements, so motor symptoms, we call those. And those abnormal involuntary movements typically are characterized mostly by what we call chorea, which is a dance-like movement. But it can also involve other things. So abnormal posture, which we call dystonia, jerky movements, or either vocalizations or some really quick jerky movements that we call tics, slurred speech, incoordination, eye movement problems, which we call ocular motor problems. Some patients even have Parkinson's-like symptoms, which is kind of the opposite of having too much movement, and even other fast movements. So abnormal movements is really one part of the disease. Psychiatric issues, or we call neuropsychiatric issues, are quite prominent as well, and they can vary widely among patients, and they kind of go the spectrum from being depressed, apathetic, to being anxious, even having psychotic symptoms like delusions, fixed false beliefs, or even seeing things sometimes, irritability, agitation, obsessive compulsive things, and even sexual issues or sleep disturbance can happen in this disease. And then lastly, but not leastly, really cognitive issues. Early on, those may be mild memory or thinking issues, slowed processing, but eventually one of the common features we see is this we call executive function, problems with planning, organizing your day, doing daily things, and needing assistance with those. As the disease progresses, unfortunately, many patients do develop a level of dementia. It can range from mild to fairly severe level of dementia as well. So symptom triad is that motor, psychiatric, and cognitive symptoms. So I wanted to kind of step back and just give you a background here about Huntington's. A lot's really happened, and I'm going to kind of bring you up to date today. That's kind of the goal of my talk here. So Huntington's disease was first described way back in the 1800s, so 1872 by George Huntington. And it took nearly 100 years for us to discover the gene. And so as you guys can see here, gene was finally isolated in 1993. And then there's this rapid progression of different discoveries that happened since then. So we've come a long, long ways. So we've got a mouse model that was developed that led to sort of understanding the disease a little bit better and what happens in the brain. And that allowed us to test things. Mouse or animal models are critical for making discoveries and lead to finding particular drugs that might be targets for treatment of Huntington's disease eventually. That led to understanding the aggregates that form in the brain, and I'll talk a little bit more about those. There's the development of research consortiums to follow patients and learn how this disease progresses. We knew very little about that, so PREDICT-HD was really instrumental. Our first clinical trial started in 2003. It was testing a over-the-counter kind of supplement called coenzyme Q10. There was also a study on creatine. Both of those actually failed, but there have been many studies since then. By the time we reached the early 2000s, 2008, the first drug was approved, tetrabenazine, for Huntington's chorea by the FDA. And now you probably know there are 3 different drugs. So there's tetrabenazine, deutetrabenazine, or Austedo, as well as more recently Ingrezza, which is generically called valbenazine, for the treatment of chorea. So huge explosion of information. Now we're into the 2010s to 2020s, and there's been an explosion of molecular and genetic information. So we've discovered things like CRISPR, which you'll hear a little bit more about in terms of gene editing. Our first gene therapy trial was this Ionis trial in 2018 that many of you may have heard about. This came out of Europe, England, testing a antisense oligo. And I'll discuss more of those targeting the Huntington disease protein. which failed, but has actually led to many, many, many more gene therapy trials. And the goal here, so far we have treatments that treat symptoms, but we don't have any disease-modifying therapies. So it's a genetic disease. So if we can fix the gene, or if we can knock down the gene product or protein product that causes disease, we all believe that we could actually either slow the disease progression or stop it in its track. And hopefully, we're all hoping that actually it might actually also improve some symptoms. Okay, so those are three different things, but I'll discuss a little bit more about those. Um, as you can see in the bottom here, there are many different trials, and I'll talk more about some of these and how they've progressed over the years. But it's amazing that just within a very short period of time, we're talking 93 from discovery, 30 plus years here, that we've gotten to the point where we have multiple trials ongoing. I will point out there are a number of these longitudinal studies that are ongoing, like Roll HD, and now we have a version 2 that's coming down the pipeline. These are really instrumental in understanding how the disease progresses and are gateways to clinical trials. And actually, many of the discoveries have come from these longitudinal studies. They're not treatment studies, but they're really critical studies for us to understand how Huntington's disease progresses over time, includes sort of your questionnaires as well as biomarkers to understand the disease. And from that, we learn a ton. So we really appreciate patients who are participating part of that. You are part of the solution. So with that, I'm going to talk a little bit more about some of the genetics and give you guys a little bit of updates. Um, I think many of you may have heard this. So Huntington's disease is a autosomal dominant disorder, meaning you only need one copy of the disease, copy of the gene, the mutant gene to cause disease. That gene was discovered a long time ago to be localized in one of your chromosomes. It happens to be localized on chromosome 4. Well, I have 22 of those chromosomes, and it was cloned back in 1993. It's actually a really big gene. It's one of 20,000 genes that we have in our body. So it's only one. And its protein product is the Huntington protein. So the problem with this gene in Huntington's disease lies within the first exon. So proteins are made of these sequences of DNA. And so DNA has the coding parts and it has junk parts called introns. Exons actually make up the protein. It happens to be the mutation occurs in the first part, the first exon, and it results in this repeat expansion. Okay. There's a normal size and there's a pathologic side, but that repeat expansion of what's called CAG, your DNA's letters are made of 4 letters, CAGT. In this case, there's a repeat expansion of CAG. You can think of it like a typewriter. We're writing out a sentence and we just get stuck on the word the, and kept typing it. So there's a repeated sequence here, and as it gets longer, that's what causes this expands me to what causes Huntington's disease, and it leads to a mutant form of the Huntington protein. So you can see that located here, and you can see how each of these repeat sequences actually codes for an amino acid that's the building block for a protein. So when it goes awry like this, shown below on the slide. Interestingly enough, at University of Florida, we have an investigator who has also shown that there are aberrant ways that genes get transcribed sometimes, what's called RAND translation, and you can get different forms of this protein. So in some cases, we get this polyglutamine, and other cases, we get different forms of the protein itself that gets expanded in the Huntington protein. So rather interesting, actually, expansion there. So what happens? We know actually a lot more about the protein when it goes awry than we know about its normal function. The mutant protein itself becomes expanded. It's easily fragmented. Turns out the exon 1 fragment is the more toxic form and more than the actual full protein. But it gets fragmented. It tends to aggregate, like in many degenerative diseases, ranging from Parkinson's to Alzheimer's disease. And those aggregates form inclusions within the cell that are actually toxic. So you can actually see pictures of those on the right. So these little blobs stained here, so they're yellow, and there's actually sort of greenish yellow, and then they're red blobs. And there's another one actually lying within the nucleus of the cell where your chromosomes are located in here. So you can imagine these little blobs like they're cancers within your body. So thinking about how the cell works, just like sort of a microcosm of your body, when you have these aggregates or abnormal proteins in the cell, like a cancer to the cell, they can cause the cell to not function properly and lead to a cascade of failures within the cell, and then the cell ends up dying. So these are actual brain cells shown here on both of these pictures. And they're not really normal, they're sick, and they eventually die, and that leads to brain degeneration. And the degeneration actually is fairly widespread, but it affects a particular part of the brain, especially, which is the striatum, or that's the main part of the basal ganglia that's important for generating movements and behaviors. Okay. Um, so I wanted to give you that picture. There's a lot more complicated things going on in the cell. There's been much written about it. I'm not going to really go through all of this, but needless to say, the fragmented protein has effects on gene expression within the nucleus. This is the nucleus of your cell. And outside the nucleus in the cell, it causes proteins to malfunction as well. And they get clogged up in those big inclusions in the cell like cancers. And that can cause different problems. So your energy production of cells gets disrupted. Transmission of signals between nerve cells, we call that synaptic dysfunction, can give you a rise to problems with thinking. And then there's transport issues that can cause as well. All of these things are part of that cascade that leads to neurodegeneration within the brain. So I'm going to take a step back again and talk a little bit about the repeat expansion. So many of you heard you're given a number typically when you're diagnosed with Huntington's disease and you've done a genetic test. So what does that number mean? I'll let Kayla talk a little bit more about that, who's our genetics counselor. But suffice to say, there's a normal repeat length, okay, and there's an intermediate repeat length that doesn't cause disease. But when that CAG repeat expansion grows to 36 or higher, you're at increased risk for developing Huntington's disease. If it's 40 repeats or higher, that risk goes to 100%. So the big question is when. People aren't born with symptoms of Huntington's disease, they develop later in life and typically within the third to fifth decades. Of course, onset of disease varies very widely. So there are juvenile onset cases, so before the age of 21, and there are late onset cases after the age of 70. We do have people who present even in their 80s with disease. So you have to probably you're already wondering, like, why are those differences? Okay. So on the right, you can see that there is sort of this graph of number of CAG repeat units and then when disease is onset. There is a correlation. It's rather weak, especially with the lower numbers. So early 40s, you can see that there's a wide degree of disease onset ranging from early to sort of in the mid 80s or 70s here. But as that repeat length goes out now, further out, or it becomes longer and longer, so folks with either 60 to 80 repeats have much earlier onset. So um, as you can see, is sort of the larger the repeat, the greater likelihood of developing symptoms early and more severe symptoms, and it may lead to what's called juvenile onset Huntington disease from that. Um, so again, I want to come back. There's been a lot of advances in trying to understand why there's this wide variability and why perhaps even a family member who has the same number or even close to the same number, or maybe even other individuals who have this number of 42 repeats, for instance, develop symptoms like in their 30s, whereas someone else doesn't present till their 80s. Well, some of that has to do with that this is only one of 20,000 genes that make up who you are. Okay, so age of onset is, although primarily determined by the length of the CAGB, it's not the whole story. There's a lot more to it. Onset of these is influenced by those other genes, as well as potentially environmental factors, and such as stressors as well. Um, Increasingly, we've also heard about this term somatic expansion. So you may ask yourself, what does that mean? Okay. So your genes are expressed in every one of your cells in your body. And we kind of think about your genetic information as the same within each cell, but that's actually really not true. So somatic differences do occur, and I'm going to just use my cats as an example here. We have four different cats at our house, so we're blessed with that, and they all look different, so that's their genetic information. You might know this type of cat. This is called a calico cat. They're all females, and they're different colors. So this yellow, this sort of orangey color, the black color here, and the white colors are due to variations in somatic expression. Somatic means body. Okay. So the variations are due to somatic mosaicism within the cells. So different genes are expressed are in the body and are in different cells. So That's what gives this cat the different colors of their skin. So this is just an example of how genes are expressed differently in the same different kind of cells from your body, but it can give varied expressions within your body. So that's not to say they're somatic. It's not the same as saying somatic expansion. So what that really means is that some of your cells they express differently, and even within different cells, there actually have been discovered over several years that some of these cells express the Huntington gene differently and may actually result in greater expansions. So that CAG expansion gene we've discovered recently is not stable among cell types. And each cell in your body may express it differently. And it turns out that we've recently learned that brain cells in particular express the gene differently and are at higher risk for developing an increased expansion. So whereas you may have CAG repeat of 42, but actually over time, we're beginning to learn that that is unstable, and actually over time, that expansion slowly gets bigger and bigger within different cell types. So there's this really interesting study that was published recently. I'm in Cell, and I can just kind of show you briefly. I'll point it out briefly. It looks pretty complicated here. What they did was they looked at patients with Huntington's disease and looked at unaffected individuals, what's called controls. So this is gene expression on the left-hand side of over here, showing different cell types within the brain. And on these normal cells, you can see the pattern is basically normal. And if you look on the right, it looks very different. I want you to focus on the red. These are cells within that striatum, that part of the brain that's primarily affected in Huntington's disease. And you can see that the numbers of those cells goes way, way down in Huntington's disease. So it turns out these are the cells that are most affected. Other cells, of course, are affected as well, but these are the ones most affected in Huntington's disease. So this study was really neat. They were able to pull those cells out, look at them in the dish, and look at the gene expression and what happens to the CAG expansion. And it turns out in different cell types in the brain that the CAG expansion grows at different rates. And it starts to grow early on, and then it starts to expand fairly quickly. On the bottom here, you can see sort of this is gene expression and the CAG repeat, and as it gets larger and larger. So this is different genes, they're different colors here, but as they reach a expansion size of over 150, the gene expression goes awry. So the proposal here really is to understand that what's going on in the body really, and this is something we're beginning to learn really, is that it's just not stable. So there are phases during your life where these genes or the CAG repeats are gradually expanding. At first it's slow, and then it starts to take off. So there's rapid expansion that occurs over years. And eventually it reaches a point where it disrupts the regular gene expression within your cells. And that's not good, okay? So some of these genes are supposed to be repressed. They're not supposed to be expressed at certain times. Other ones are supposed to be active and they get repressed. So this aberrant gene expression within your cells becomes altered at a point where we have a CAG expansion of over 150. So I think the first thing I want you to take away here is that, yes, we do talk about the CAG repeat number that you get when you get diagnosed, and that's important. It does increase your risk, but what's going on in the body and the brain is that it's not stable. And within the brain cells, it's actually increasing over time, and that we get expansions that are over 150, or even higher. And this may explain some of our animal models. Turns out when we make animal models, it's required to have animal models with CAG repeats of over 100, even 150, to develop any symptoms. So we're sort of now just realizing, wow, based on this information, this is really interesting. What's also really critical to understand is that it supports evidence that there are modifier genes in our body that control this expansion as well. And that maybe targeting these modifier genes, all those other 20,000 genes that you have in your body, may be a way to treat or even slow down this gene expansion and maybe even stop the onset of disease. So we've been focused up to date on the Huntington genes, but we may now start to focus on other genes that are regulating this cell process. And it has widespread therapeutic implications for other diseases that have similar mechanisms of gene expansion. So Really interesting stuff. I know it's more advanced, but this is key to helping us as scientists to develop new gene therapies to hopefully treat disease. So I'm going to turn a little bit to talk briefly about some of the things that are going on. I know it's getting confusing. You have DNA that codes RNA, and then the RNA is kind of the instruction thing that carries outside this nucleus and then tells you what proteins to do. So we have all kinds of things going on in our cells. So I'm going to try to put them together. But suffice to say that DNA is that instruction set. To go from DNA to proteins, you need to read the DNA with them. And that the way we read is by creating something called RNA or messenger RNA that encodes the building blocks, which are amino acids that make up proteins. And if any part of this goes awry, you get a bad cake, basically. Right? So gene therapy in general is really exciting, holds great promise, and for novel treatments, um, just know that there are many stages where we can target therapies. It'd be great if we could do it at the DNA level. We're not quite there, or we're getting really closer, but there are all different spots where we can actually target it. Many ways to manipulate genes. We can either increase their function or decrease their function. Some genes cause deficits by a gain of function, in other words, a property that causes symptoms. Some genes, because they aren't properly expressed, actually cause a decrease of function as well. And some are mixed actually as well. So to do gene therapy, we need to fix the gene or we need to induce a modifier gene or express perhaps a defective gene to help it. So in the case of Huntington's disease, there are mixed effects. The main way that we think that we can do gene therapy is by silencing or knocking down the mutant Huntington protein itself. There are lots of ways to do this, okay? So, and this is just a schematic. This blue portion, this blue box is your nucleus with your DNA and your chromosome. And then outside that is the cell area, okay? So we can target the DNA by using CRISPR. You may have heard of CRISPR, and there are other techniques that are there. We can target things by other inhibitors. We have things that are called antisense oligos. These are little snippets of DNA. They're usually fairly small. Um, they're pretty stable, but they do get degraded. They target mRNA, okay, pre-RNA that goes out into the cell and then starts to encode some of your proteins. So this was an example of this was the Generation HD trial used oligos. These have to be injected into the body um, so they can circulate around the brain. Okay, so they can't be taken orally. Okay. There are other forms of genetic therapy which are viral, and I'll talk briefly about that. Antibodies have been looked at. These are proteins, and you can use vaccines, or we can passively immunize somebody by injecting or infusing antibodies. Not something that's been used very much yet for Huntington's. And then there are other different forms we call small molecule inhibitors. I'll talk a little bit more. But ultimately, the goal is to reduce the production of the mutant Huntington protein and those formation of those toxic aggregates. Okay. So delivery is the big issue here. How do we deliver gene therapy? And I've kind of mentioned a few of those. Some of them we have to inject. Some of them may be in form of pills. There are other vehicles that we use, like these small liposomes, which don't cause any kind of immune reactions. And then sometimes we use viruses. These viruses have been engineered not to cause pathology. We typically, the one that used most often is called the adeno-associated virus, which is related to adenovirus, which causes the common cold, one cause of the common cold. So it's been genetically engineered not to do that, okay? But it's a really great vehicle to deliver gene therapy in a very, very safe way to different parts of the body, including the brain. And we're here to talk about that briefly. So we've come a long way with gene therapy, and I'll just go over a few of the studies that are ongoing. So I mentioned Generation HD, and then there's the what's called HD2 that's just finishing up now. So the first set, this was the Ionis trial that was then bought up by Roche and Genentech. This involved those ASOs or antisense oligos that were injected into the spine, and we had over 600 patients who actually participated in this trial. There were some issues with it, but generally it did not meet its endpoint. In reviewing the data, the companies actually noticed that perhaps really early onset patients or younger patients actually may have shown some benefit. And so they started this version 2 trial, And so we've been doing this trial along with a company as well called Generation HD2, and we're hoping to see if there's those results. So that's coming out. Um, Sparks Therapeutics is the division, I believe, of Roche as well, and they're also doing gene therapy to lower mutant Huntington. You'll hear more about this. They have done what's called a first phase trial as well as a phase 2 and 3 study. This is actually limited to Australia and New Zealand. Um, Wait to hear from this. They've already got about 102 patients, 120 patients, or they're trying to get that number. If successful, it could come to either Europe or the United States eventually. But that's another gene therapy trial that's ongoing. So I'm going to turn just briefly to the AMT 130 results. So most of you probably heard the press release that was out. This was actually a trial sponsored by Unicure. And so this press release came out of Europe. It was extremely exciting. So let me go over this trial very briefly. This was a phase 1, phase 2 trial, so very early onset trial. And the gene therapy that was used here was a knockdown approach, and it was using a virus to deliver that into the brain directly. So this particular trial actually involved several brain injections and a brain surgery. So it's a quite involved trial. It actually involves 6 injections in the brain that takes about a half a day. So it's a very involved surgery. And the target population was very early onset Huntington's patients who had very little, if no, degenerative brain issues. So at this point, roughly 29 patients have been enrolled. They did 2 doses of both a low dose and a high dose. And what was really exciting about this is they recently had this press release that's showing that it was successful in lowering what was called the composite UHDRS score by 70% compared to a control group. Now, there are a couple of limitations here. The benefits were only seen in the high dose group, not the low dose group. And actually, it says 17 or 19, actually 17 patients. Only 12 of those actually completed the 36 months. So it's even smaller group we're talking about. The low dose group did not show any benefit. So the composite UHDRS scale is actually a way to measure both the motor, cognitive, as well as psychiatric issues in a Huntington's disease patient. It sounds big, but it's just a sort of a separation in the curve. One of the major limitations of this trial, in most trials, we typically like to have a control group, and most trials are randomized. This was not really randomized. And the control group here was unethical. So we couldn't really do a sham, so surgery in patients with Huntington's disease. So again, that was felt to be unethical. So there was a big issue in terms of what would be the comparison group. So only a small portion of patients actually in the first cohort had what was called a mock surgery. They undergo sort of the surgical procedure, but they didn't really get anything injected. Okay, but that involved only 10 patients. The majority of the patients were actually compared to what's called the historical control based on either enroll or predict HD patients. So that's not a very ideal control, but again, that's a very established control group, and we know how they progress, but definitely a limitation. So I want to point out some of those limitations of the study. It's a very small study, and the control group's kind of not a direct comparison group. But regardless of that, this is a really exciting thing. Any kind of success in Huntington's disease is really exciting because we've had none so far. And not only did they see changes, positive changes in the what's called composite UHDRS score, they also saw changes in what's called TFC, which stands for total functional capacity. So that means your functional abilities. And they saw favorable trends in other cognitive scores, as well as one brain biomarker called neurofilament light. So there are multiple positives from this study, which really demonstrate the proof of principle that gene therapy for Huntington's disease may actually be very helpful. And again, this was very well tolerated. Yes, they did have some safety issues, but they have been able to overcome them and mitigate those issues for the surgery. So very exciting. Unicure does plan to submit this for an expedited approvals from the FDA, but I will tell you there's probably a lot of hurdles for them to go. So they do expect to have approval by the end of the year, but I think that's a fairly long shot. It's very likely that the FDA will have lots of questions for them. They may even require them to do further studies. So I've listed a few of those hurdles. This company will have to be able to manufacture gene therapy at scale. The surgical issues are limiting. Many patients may not want to do that. It's kind of scary to think about that. So who's going to be the right patient for this? And will it be limited to a very small population? And then finally, you have to think about costs of things. This could be quite expensive as a therapy because it does require a brain surgery. So there are a lot of other trials ongoing. This is really exciting. So I don't want to, want too much. I'm going to show you a few other things, many more trials ongoing. Um, so some of these trials don't involve brain surgery. So these are oral drugs. So these are small molecule inhibitors, and there's several of them. So branaclam was the first one, and it failed because it caused neuropathy. But there's another trial upcoming, probably in the next year, called for on voltaplam. This was initially developed by PTC Therapeutics, but it's being taken over by Novartis. So there have been positive results in a phase two study, and so they're planning a phase three trial. So for those of you interested in research, this is the next one coming. Skyhawk is also developing a similar kind of splicing drug that's oral. Oral is much better than having injected in the brain. This is ongoing outside the US, Australia, New Zealand, but may come to the United States. Those of you who participated in the perdopidine, that's another oral drug, not necessarily disease modifying, but may slow progression. Although the primary endpoint was not met, it seemed favorable and a very well tolerated drug. There are other gene therapies as well. I mentioned the ASOs, like for generation. I'll come back to those. There are two other companies called Vico and there's Wave. Wave Neurosciences or Life Sciences is actually unique in the sense their ASO actually targets specifically the mutant Huntington protein, whereas all the other ones target all Huntington. So listen and pay attention to see whether Wave comes out. They're targeting ASO, may be limited to a specific population as well. And there are other trials coming along the way. So it's a really exciting time for folks. I'm going to finish there and just kind of summarize saying that this is a complex disease with lots of different features to it. We need new treatments. We definitely need a disease modifying therapy. Genetics is complicated, but we're learning a lot more. It's a lot more complicated than we thought than just the CAG repeat. And this discovery of expansions, somatic expansions, is huge. Treatment may be require what we call multimodal and team approach, of course, but there are novel therapies coming down the pipeline. So I'm really excited. I'm going to stop there and let my colleagues take over. I'll be available later on today, and we can please do put in the chat other questions for us, and we'll try to answer those a little bit later today. All right. Thank you all.